On May 19th, 2016, a research team led by geneticist Andres Ruiz Linares published a paper in the journal Nature Communications that would quietly rewrite what science understood about one of the most overlooked parts of the human face. The paper looked ordinary on the surface. It was a genome-wide association study, the kind of dense academic document that almost never breaks out of specialist circles, but buried inside more than 6,000 genetic profiles collected from people across Latin America, was a discovery that would eventually connect a single physical feature, the width and shape of the human nose, to a lost continent, an extinct species of human, and a migration story that stretches back over 50,000 years. If you have ever looked in the mirror and wondered why your nose is broader than a friend's, or flatter, or wider at the base, you are about to learn that the answer has almost nothing to do with random chance. It was written into your ancestors' bodies by ice ages, by land bridges that no longer exist, and in some cases, by interbreeding with a species of human that vanished from the planet tens of thousands of years ago. And if you are Filipino, Indonesian, Malaysian, Pacific Islander, Melanesian, Southeast Asian, or carry indigenous ancestry from the Americas, this story is about you directly, because the traits at the center of it are running through your genome right now. Hit subscribe, because every week we take a deep dive into DNA research, ancestry mysteries, and the science that is only now starting to explain where our physical features actually come from. Let's start with what that 2016 study actually found, because most people who have heard fragments of this story only know the headline version, and the headline version leaves out almost everything that makes it interesting. The research team, working with the Candela cohort, a large genetic database built specifically to study mixed populations across Mexico, Colombia, Peru, Chile, and Brazil, set out to answer a question that had barely been studied before. Earlier research into the genetics of facial structure had focused almost entirely on European populations, which, compared to the rest of the world, show relatively little variation in nose shape. By studying a population with a much wider range of ancestral backgrounds, the researchers were able to isolate genes that simply hadn't shown up in earlier work. They didn't treat nose shape as one single trait. They broke it into specific, measurable components. The angle of the columella, which is the strip of tissue between the nostrils, the breadth of the nasal bridge, the width of the nostril wings, and the overall pointiness or flatness of the nose tip. Then, they matched each of those measurements against the genome of every participant, searching for statistically significant links. 4 genes stood out. The first was DCHS2, a gene involved in cartilage growth and craniofacial development, which turned out to be the strongest predictor of nose tip pointiness. The second was RUNX2, a gene that had already been studied for decades because of its role in bone formation, but had never been tied to nose shape until this study connected it directly to the width of the nasal bridge. The third was GLI3, which showed the strongest association with nostril breadth. And the fourth was PAX1, which also influenced how wide or narrow the nostrils sat on the face. All 4 of these genes are involved in the same basic biological process, the way cartilage and bone form and grow during fetal development, and all 4 of them showed clear differences in frequency across different ancestral populations. People with certain DCHS2 variants tend to have a narrower, more projected nose tip. People with the alternate variant tend to have a flatter, broader profile. RUNX2 variants linked to a wider nasal bridge turned out to be common in populations with tropical and equatorial ancestry, while variants linked to a narrower bridge were far more common in populations from colder, drier parts of the world. At first glance, that pattern lines up perfectly with something scientists have suspected since at least the 1970s, an idea called the climate adaptation hypothesis. The logic is straightforward. In hot, humid environments, a wider nose with larger nostrils allows more air to move through without much resistance, which is exactly what you want when the surrounding air is already warm and moist. In cold, dry environments, a narrower nose forces incoming air through a longer, more constricted passage, which warms and humidifies it before it reaches the lungs, protecting delicate lung tissue from cold, dry air damage. This correlation between climate and nose shape is well documented, strong enough that forensic anthropologists have used nasal measurements for decades as one clue when estimating a person's geographic ancestry from skeletal remains. On the surface, it looks like a tidy story. Hot climates produce broad noses, cold climates produce narrow ones. Evolution slowly sorts everyone into the shape that suits their environment. Except the moment researchers looked closely at where these gene variants actually show up around the world, the tidy story fell apart. The broad nose variants linked to RUNX2 and DCHS2 are common, as expected, throughout sub-Saharan Africa, Southeast Asia, and Melanesia, all classically warm, humid regions. But they also turn up at strikingly high frequencies among indigenous populations in the high-altitude Andes of South America, a cold, dry, oxygen-thin environment that should, according to the climate model, favor narrow noses. They turn up in parts of East Asia far from the equator. And when researchers ran the numbers on how fast these variants appear to have spread through certain populations, the timeline didn't fit slow, gradual climate selection stretched across 100,000 years. Something moved faster than that. Something concentrated these variants in specific groups of people within a much narrower window of time, and climate alone could not explain it. That puzzle is what pushed researchers toward an entirely different field: ancient DNA. Over the last 15 years or so, the ability to extract and sequence genetic material from human remains that are tens of thousands of years old has completely transformed what we know about human prehistory, and nowhere has that transformation been more dramatic than in Southeast Asia. For most of the 20th century, the accepted model of Southeast Asian population history went something like this. An ancient group of hunter-gatherers, sometimes referred to as Australo-Melanesian in older literature, settled the region first, and their descendants today are represented by groups like Aboriginal Australians, Papuans, and some indigenous communities in the Philippines and Malaysia. Then, roughly 4,000 to 5,000 years ago, a large wave of Neolithic farmers moved south out of what is now southern China and Taiwan, bringing agriculture, new languages, and new tools with them. Over time, these farmers either replaced or absorbed the earlier hunter-gatherer populations so thoroughly that the original genetic signature nearly disappeared. This framework is known among linguists and archaeologists as the out-of-Taiwan model for the spread of Austronesian languages, and it explained a lot. It accounted for why Austronesian languages are spoken across an almost unbelievable geographic range, from Madagascar off the coast of Africa all the way to Easter Island in the middle of the Pacific, and it lined up with the archaeological record of agriculture, spreading through the region. Then ancient genomes started coming back from sites in Vietnam, Laos, and Malaysia, some of them 8,000 years old or more, and they did not match either half of the expected picture. These early inhabitants weren't simply the ancestors of Aboriginal Australians and Papuans, and they weren't simply an early version of the farmers who would later arrive from the north. They were something else, a genetically distinct population carrying its own unique signature, one that included a very high frequency of the broad-nose variants identified decades later in the genetic association studies. Researchers now often refer to this population using the term Sundaland, named after Sundaland itself, the vast landmass that once connected mainland Southeast Asia to what are today the islands of Sumatra, Java, Borneo, and much of the Philippine archipelago. During the last glacial maximum, roughly 20,000 years ago, so much of the planet's water was locked up in ice sheets that global sea levels sat more than 100 meters lower than they do today. Sundaland, during that period, was not a scattering of islands. It was a single continuous continent, larger than the entire landmass of Western Europe, crisscrossed by rivers, covered in forests and grasslands, and fully capable of supporting a large, stable human population. Ancient DNA evidence now suggests that this Sundaland population persisted for tens of thousands of years, quite possibly 50,000 years or more, as a genetically distinct, well-established group, not a temporary band of migrants passing through on their way somewhere else. When rising sea levels at the end of the Ice Age gradually swallowed Sundaland, turning a continent into an archipelago, its people didn't vanish. And when Neolithic farmers began arriving from the north a few thousand years later, they didn't simply erase what came before them. They mixed with the existing Sundaland population, and the degree of mixing varied enormously depending on the region. On the Southeast Asian mainland in places like Vietnam, Thailand, Cambodia, and Laos, the blending was substantial, and today's populations there carry both ancestries in roughly comparable proportions. In the islands, the picture is even more layered. The Philippines in particular shows evidence of at least 3 or 4 distinct ancestral migration waves stacked on top of each other over thousands of years, each one contributing a different genetic thread to the population that exists there today. But running through every single one of those layers, present in every wave of migration and every mixing event, are the broad-nosed gene variants, because they were already present in the original Sundaland population before any of the later migrations ever happened. And they simply rode along through every subsequent generation. That explains a lot of what we see across island Southeast Asia. It does not, by itself, explain why the same variants show up in parts of East Asia far from any tropical coastline, or why they appear at meaningful frequencies among indigenous populations in South America. For that piece of the puzzle, we have to go back even further, to a population researchers sometimes describe using the term Ancestral East Asian lineage, a group that predates Sundaland itself and that eventually gave rise to 2 very different branches of humanity. One branch moved south and became the ancestors of the Sundaland population. The other moved north, eventually becoming the ancestors of Northeast Asian populations, and critically, the ancestors of the small groups who crossed from Siberia into the Americas across the Bering Land Bridge sometime between roughly 15 and 20,000 years ago. This detail matters enormously, because it means the founding populations of the entire Western Hemisphere descended from this same ancient East Asian lineage, and they carried the broad-nosed gene variants with them across that land bridge. That is the real explanation for why indigenous peoples across the high Andes, the North American plains, and the Amazon rainforest show a noticeably higher frequency of these variants than the climate adaptation model alone would ever predict. They didn't develop broad noses because of the environments they eventually settled in. They inherited them, already present, from tens of thousands of years of ancestry that traces back through ancient East Asia, and before that, back into Sundaland. This has real, practical consequences for how modern commercial ancestry testing works, and it's a problem that geneticists have been quietly raising for years. The reference panels used by major consumer DNA testing companies, the databases they compare your genome against to generate those ancestry percentage breakdowns, are built primarily from contemporary populations, sampled in relatively recent decades. Those panels were not built with the full 50,000-year depth of Sundaland ancestry baked into them, and they were not built with the tens of thousands of years of genetic drift that shaped Indigenous American populations after the land bridge crossing. The practical result is that broad nose variants and the broader ancestral signatures connected to them in people with Filipino, Indonesian, Pacific Islander, or Indigenous American backgrounds often get flattened, mislabeled, or underrepresented in commercial ancestry reports. It isn't that the genetics aren't there. It's that the comparison data isn't deep enough yet to read them with real precision. Research groups like the Reich Lab at Harvard and large-scale international efforts, such as the Genome Asia 100K project, have spent the last several years specifically trying to close that gap, adding ancient DNA and long underrepresented modern populations into their datasets, because the existing commercial panels simply weren't capturing enough. Now, here is where the story takes its strangest turn, because no shape in ancestry in this part of the world cannot be fully explained without talking about a species of human that went extinct tens of thousands of years ago, and that we only learned existed a little over a decade ago. In 2010, researchers sequenced a genome from a tiny fingerbone fragment discovered in Denisovan cave in southern Siberia. There was no skull, no substantial skeleton, just that fingerbone, a couple of teeth, and a remarkably complete genome extracted from that scrap of ancient material. That genome belonged to a previously unknown group of archaic humans now called Denisovans, a population closely related to Neanderthals, but genetically distinct from them. When scientists compared the Denisovan genome against modern human populations around the world, they found something nobody had anticipated. Denisovan DNA survives in modern humans today, but it is not spread evenly across the globe. It is overwhelmingly concentrated in one part of the world, Melanesia. Populations in Papua New Guinea, the Solomon Islands, and Vanuatu carry somewhere between 3 and 5% Denisovan-derived DNA in their genomes, a larger fraction than the Neanderthal ancestry found in most European or East Asian populations. In 2021, an even more specific discovery emerged when researchers studying the Aita Magbukon, an indigenous group in the Philippines, found that this particular population carries the highest documented level of Denisovan ancestry of any group on Earth, edging out even Papuan populations, evidence of a separate, distinct pulse of interbreeding that happened somewhere in island Southeast Asia rather than only in New Guinea. That same year, a separate genetic study published in Science Advances, again working with the large Latin American Candela cohort, identified a specific region of the genome, one that had entered the modern human gene pool through Denisovan introgression that appears to influence facial shape and structure. That finding matters because it means some portion of the facial variation we see in modern populations with deep Melanesian and Southeast Asian ancestry isn't the product of modern human evolution at all. It came from a completely different, now extinct branch of the human family tree, one that evolved in isolation in Asia for hundreds of thousands of years before modern humans ever arrived and began interbreeding with them. When you look at someone with strong Melanesian or Southeast Asian ancestry, some fraction of what you're seeing in their facial structure may trace back not 50,000 years, but potentially 300,000 or 400,000 years to a lineage of humanity that no longer exists anywhere on the planet, except as fragments preserved inside the DNA of their living descendants. It's worth pausing here to be precise about what the science does and does not claim, because this is exactly the kind of story that tends to get exaggerated the further it travels from the original research. The Denisovan-linked genomic region identified in that 2021 study is associated with general facial variation, not a single isolated nose gene working in total independence from everything else. Nose shape itself is polygenic, meaning it is shaped by the combined interacting influence of dozens of different genes, several of which we've already discussed, DCHS2, RUNX2, GLI3, and PAX1 among them, plus additional loci researchers have identified more recently in populations across East Asia and Europe. None of those genes act alone, and none of them map to a single ancestry group in any simple one-to-one way. A person can carry a DNA test result showing 70% one ancestry and 30% another, and still have no straightforward way to say which side of that ancestry gave them their nose shape, because the genes influencing that single feature are frequently inherited from multiple different ancestral lines at once, recombined and passed down independently of each other across generations. What the research does establish, with real confidence, is the bigger pattern. Nose shape across human populations is not random. It reflects a layered history of climate adaptation working over hundreds of generations, deep ancestral population structure inherited from groups like the Sundaland population and the ancient East Asian lineage that came before it, and in some populations, direct genetic inheritance from an extinct species of archaic human that modern humans encountered and interbred with tens of thousands of years ago. Every one of those forces left a measurable trace, and every one of those traces is still sitting in the genome of people alive today. There is one more evolutionary force worth mentioning, because people frequently ask about it: sexual selection. The idea that mate preference for certain physical traits can shift how common those traits become in a population over time. It's a real, well-documented evolutionary mechanism, but its specific role in shaping nose morphology remains genuinely unsettled among researchers. Some scientists argue it played, at most, a minor supporting role behind climate adaptation and ancestral population structure. Others argue that in smaller, more isolated populations, where mate choice can shift the frequency of a visible trait relatively quickly, sexual selection may have reinforced and sharpened differences that climate adaptation had already started. The honest answer, and the one most researchers would give you directly, is that we don't fully know. By the time ancient DNA lets us observe these populations at all, the broad-nose variants are already present at high frequency, and untangling exactly how much of that early frequency came from climate, how much from population history, and how much from mate preference is still an open and actively studied question. It's also worth noting that these same four genes, DCHS2, RUNX2, GLI3, and PAX1, show something else in the genetic record besides population-level frequency differences. They carry strong signals of recent positive selection in modern humans, meaning that at some point in our evolutionary past, the pressure to have one version of these genes over another was strong enough to leave a clear statistical fingerprint, distinguishing modern human variants from the equivalent regions found in Neanderthal and Denisovan genomes. GLI3 in particular appears to have gone through unusually rapid evolutionary change, suggesting nose and nostril shape were under real evolutionary pressure rather than simply drifting from one generation to the next. Follow-up research since then, including studies on Han Chinese and European cohorts, has confirmed associations with RUNX2 and PAX1, while turning up additional loci not captured in the original 2016 analysis, a reminder that this field is still expanding rather than closed, with every new population adding a slightly different piece to the picture. Now let's bring this back to something a little more personal, because the genetics here isn't just an abstract academic curiosity. If you carry Filipino, Indonesian, Malaysian, Pacific Islander, Melanesian, broader Southeast Asian, or Indigenous American ancestry, and you have a broader or flatter nose shape, you are, in a very literal and documented sense, carrying genetic material from a population that survived on a continent that no longer exists on any modern map. You may be carrying DNA passed down from interbreeding with an entirely separate species of ancient human that vanished from the planet tens of thousands of years ago. And you are connected through an unbroken chain of ancestry stretching back through ice ages and land bridges to a group of people whose descendants eventually spread across roughly half the surface of the Earth. That is not a small thing to carry in your face. There's a cultural dimension to this story that deserves attention too, especially for people who grew up in Western countries while carrying Southeast Asian or Pacific Islander ancestry. For much of the 20th century, broader facial features associated with Asian and Pacific populations were often culturally devalued against Western beauty standards, sometimes by outside cultures, and sometimes internalized within the very communities carrying those features. Cosmetic rhinoplasty rates in parts of East and Southeast Asia, particularly in South Korea, Japan, and the Philippines, are among the highest in the world, and the most commonly requested changes involve narrowing the nasal bridge and reducing nostril width, the exact traits that trace back through this deep ancestral history. Genetics doesn't tell anyone what to do with their own face. Personal choices about appearance are exactly that, personal. But understanding where a trait actually comes from, understanding that it represents 50,000 years or more of survival, migration, and adaptation, changes the way that information sits with a person. It's harder to view something as a flaw once you understand what it took to still be here carrying it. Commercial ancestry testing is only going to get better at capturing this complexity over time. The reference panels being built today by academic research groups are steadily incorporating more ancient genomes and more historically underrepresented living populations, specifically because current commercial databases are still missing significant genetic depth in exactly the regions this story covers. Researchers strongly suspect there are additional archaic populations beyond Neanderthals and Denisovans that also contributed DNA to Southeast Asian and Pacific genomes and that haven't been fully characterized yet. Every year, more ancient genomes get sequenced, more ancient population structures get identified, and the picture of Sundaland and the peopling of Southeast Asia and the Pacific gets sharper and more detailed. Ancestry reports handed to Filipino, Indonesian, Pacific Islander, and Indigenous American customers 5 years from now are almost certainly going to look meaningfully different from what those same companies produce today, with more layers, more nuance, and a fuller accounting of exactly how deep these lineages actually run. If this kind of research interests you, and if you've made it this far, it clearly does, subscribe and turn notifications on, because we go deep into topics like this every single week, taking real peer-reviewed findings and explaining what they actually mean for real people trying to understand their own ancestry. And if there's a specific ancestry question you want us to dig into next, leave it in the comments below. We read all of them, and a lot of our upcoming episodes get built directly around the questions that keep showing up. Before we go, it's worth addressing something that comes up almost every time this topic gets discussed, because it trips people up constantly. What ancestry percentages on a DNA test actually mean when it comes to physical traits like nose shape. A lot of people assume that if a test says there's 70% one ancestry and 30% another, any single visible trait like nose width must obviously trace back to whichever ancestry is the larger percentage. That is not how the underlying genetics actually works. A trait like nose shape is influenced by multiple separate genes, and each of those genes can be inherited independently from different ancestral lines, recombining generation after generation in ways that make simple single-source attribution essentially impossible. What research has established is probabilistic, not deterministic. In cold, dry climates, a narrower nasal passage with greater internal surface area allows incoming air to be warmed and humidified more effectively before it reaches the lungs, protecting delicate respiratory tissue from damage. In hot, humid climates, that same narrow structure becomes unnecessary and even mildly disadvantageous, so a broader, more open structure was favored instead. Those small survival advantages, repeated across hundreds of generations, are more than enough to reshape the genetic frequency of an entire population over tens of thousands of years. Every nose shape found anywhere on Earth today represents a functional adaptation to something. None of them are biologically superior or inferior to any other. They are simply better or less well matched to the specific environments and the specific ancestral histories that produced them. And the genetics behind that matching persists long after the environment itself changes, carried quietly forward, generation after generation, written into the shape of every face that inherits it.